Hi, this is a video to introduce you to the Plug and Play Cancer Genomics project. So while completing this project, you are going to be conducting biomedical research. So starting off big picture, if you're doing biomedical research, there's multiple different ways that you can approach a problem. So first, you can do something called an in vivo approach, which would be examining a problem looking at the whole organism. So this could be a mouse experiment, human clinical trials. Alternatively, you can do something called an in vitro approach. So this would be studying a problem by looking at something on the cellular level or even parts of cells. So something outside of their biological context. And then lastly, you can do what is called an in silico approach. So in silico biomedical research is involved with using computers or computer simulations in order to answer biological research questions. So as doing this project, that's the type of research you're going to be doing. And we're going to be using a resource called the Cancer Genome Atlas. So the Cancer Genome Atlas is a massive online repository that contains genomic data from cancer patients. Primarily, these data are going to be something called RNA-seq, which is short for RNA sequencing, and I'll explain what that is in just a minute. So this is all genomics data taken from cancer patients, and these samples were taken through biopsies, and they were collected from patients with HIPAA and IRB considerations in mind in order to get patient consent and then also protect their privacy as well. So once these samples were collected and analyzed, all of the data is uploaded onto the Cancer Genome Atlas, and now these data are freely available for anybody to analyze. So as mentioned, the type of data that we're going to be looking at is something called RNA-seq, again, short for RNA sequencing. And the whole point of an RNA sequencing experiment is to collect the RNA from different biological samples and then do various comparisons depending on your experimental samples. So in the case of cancer biology research, usually you're going to be looking at RNA taken from tumor biopsies, and you might be comparing that to a non-tumorous biopsy to look at the difference in the genetics. So after you've identified your biological samples, you'll then collect the RNA. And there's different ways that you can do this depending on your experimental goals. So for example, if you're interested in gene expression, you might just be collecting the mRNAs. Whereas if you're more interested in looking at maybe non-coding RNAs, you might collect total RNAs from all of your cells or tissues. After collecting your RNAs, you will create a cDNA library with reverse transcriptase to stabilize the samples. And then that will be sent off to a sequencing facilities. And they will do the sequencing, and you will get back all of the A's, T's, G's, and C's from all of your samples. So you're left with this massive data set. But with that data set, you can do comparisons to examine different experimental questions. So just to give you a few examples, you might be interested in what are the different types and levels of mRNAs. So this is a common approach called differential gene expression. Alternatively, let's say you had done a total RNA collection, you might be able to examine some of the differences in non-coding RNAs. So your goals for this project is you are going to use data from the Cancer Genome Atlas in order to investigate a question in cancer biology. So I'm providing you with template questions so you can customize your research to let it fit your interest. Once you've selected your data, you're going to use an online tool called cBioPortal. So this is web-based and free to let you analyze those data. You're then going to perform a literature search and create an annotated bibliography. This will be a crucial step for explaining the why of the data you collected, and I'll talk about that a little bit more in a bit. And this will culminate in you creating and presenting a scientific poster. So as mentioned, I'm going to provide you with a guide with template research questions. So these all are designed so that you can customize those and investigate something with cBioPortal that is related to your interest. So the first guide will tell you about collecting data if you want to look at which genes are going to predict survivability in a cancer of your choice. Secondly, you can look at genetic differences in different grade tumors. And then thirdly, you can investigate the impact of a particular demographic factor, such as race, ethnicity, age, and sex, on patient survivability in a cancer of your choice. There is also a fourth create your own question option, but in order to do this one, you should meet with your instructor to make sure that they have approved your experimental plan. So you will follow the guide for these template research questions to collect your data from CBio Portal. So I wanted to briefly talk about some of the types of data that you'll collect from CBio Portal. So all three template questions, you will be asked to collect what are called Kaplan-Meier survival graphs. And if you're unfamiliar with these, you will see percent survival on the y-axis and time on the x. And the lines will show you what percentage of patients are still alive at a given time point. So you can break data apart and then see if different parameters influence patient survivability. So in the case of this example, we see patients that have gene A signature have better survivability than those with gene B signature. And then you can do statistical comparisons of your groups to see if anything is significant. So speaking of statistics, what is lovely about CBio Portal is that it will do all of the statistics for you. So as you're generating these graphs, CBio Portal will calculate the p-values for you, so you can see if any of those differences are significant. So a p-value less than 0.05. Additionally, depending if you have a sufficiently large data set, CBio Portal can also calculate hazard ratios for you. So hazard ratios can be calculated in various ways, but in this context, you're looking at death in the treatment group divided by death in the control group. So if you do this calculation and you have a hazard ratio that's 1 or close to 1, it says that death in both groups is pretty much the same. However, if you do this calculation and your number is much greater than 1, then it indicates that the treatment group has more death, and therefore we would say it's more hazardous, so they have weaker survival. Conversely, if you do this calculation and your value is less than 1, then it means that there's more death in your control than treatment, and your treatment is then considered less hazardous, and those patients will be more likely to survive. So to show you a quick example of that, this is data collected by a former student of mine, where she was looking at the impact of age on glioma patient survivability. So her control is the younger population, and her experimental are these older age groups. And in each case, there is a higher hazard ratio, indicating that the older a patient is, the more hazard it is, and the less likely they are to survive. If you do template research question 1, you'll be collecting something called an oncoprint. So an oncoprint is a pictorial representation of mutations in a particular gene in a patient data set. So as an example, we're looking at an oncoprint, again, created by a former student of mine, looking at APC and TP53 in colorectal cancer patients. So if you're looking at this, you will see numbers listed next to the gene name. That tells you how frequently those genes are altered in a particular data set. Additionally, along the oncoprint, you will see each sliver is an individual sample, and then those samples are color-coded based on the type of mutation. So we see in this particular data set a lot of truncation mutations and missense mutations. So very quickly, you get this nice graphic representation of the genetic alterations in that particular gene. If you're using template question 2 or 3, you're going to be collecting something called a genetic alterations graph. So this will build a graph that shows you the top 10 most altered genes in a particular data set. As an example, some more data from a former student. This is looking at mutations in head and neck cancer patient data set. And these are the top 10 most altered genes divided by race. All right, a little bit of a deviation now. So you're going to go to CBioPortal, and CBioPortal does a lot of the heavy lifting for you because it does your stats. It makes beautiful graphs for you. But a big part of any type of genomics, bioinformatics research is giving the context for your data. So when you're looking at your graphs, you might see that, oh, this gene, the patients do better, or these genes are differently expressed in higher grade versus lower grade tumors. But while CBioPortal can do the graphs, do all the statistics for you, it's still up to you to explain and interpret that data. So contextualizing your data, and just as a great story that goes along with that. If you're unfamiliar, this is from a book series called The Hitchhiker's Guide to the Galaxies. They're classics. They're amazing. Read them if you haven't. But in this particular story, we have an advanced civilization that builds a supercomputer. That's deep thought there. And they build a computer to answer the question, what is the meaning of life? And they have this massive supercomputer and it thinks on this question. And after a period of time, it spits back the answer 42, which is the data from the computer, but the actual meaning and of that is lost on the people. So that's the rest of the books. They're trying to figure out what is the meaning of 42. So CBio portal data and bioinformatics data in general can be very similar. The computer spits numbers out at you, but it's really up to you to be able to explain the how and why. So with that, once you have your data from CBioPortal, you are going to be building an annotated bibliography. So you need to go to the literature and read about those genes that you've identified. Think about what they do on a cellular level. Based on what they're doing on a cellular level, why might that impact patient survivability? So you'll build your annotated bibliography, and I have a link here, but I'll also put it in the video description. If you want more resources on building an annotated bibliography, I recommend the Purdue Online Writing Lab. After you've done your literature search, you will write an abstract, put your data together, and compile your project into a scientific poster. And I will provide you with a template for generating this poster. I also really recommend this video, which I'll also link in the description, how to create a better research poster in less time. So this project is not meant to be done all at once, so it's divided into several checkpoints. So whether you're collecting your data from CBioPortal, your annotated bibliography, making your poster, and your final presentation. So make sure to check with your instructor for specifics on due dates and how to submit these. So thanks for listening, and I hope you have a good time doing this project.